For our last talk today, we're moving back to the bedside. And, several heard today already about the toxicities of CAR T cells. Certainly one of the most complicated ones is neurotoxicity. Cynthia Harrison, who is now a medical student, she, I think, has been a postbac until recently in the Pediatric Oncology branch, working with Dr. Nurali Shah's team, but under primary mentorship of Dr. Hanin Shalabi, who is here today and who is a leader in the leukemia team, is going to talk about approaches to treat acute neurotoxicity in pediatric patients getting CAR T-cell therapy. The title of her talk, Intrathecal Hydrocortisone for Treatment of Children and Young Adults with CAR T-cell Immune-Effector Cell-Associated Neurotoxicity Syndrome. Thank you for talking to us and congratulations.
Hello, everyone. Oh, perfect. So my name is Cynthia Harrison. As Dr. Wiedemann said, I recently finished a post-baccalaureate research year at the NIH Pediatric Oncology branch under Dr. Hanin Shalabi, and I'll be starting medical school at NYU Grossman this summer. And I'm very excited to be speaking with you about I'm very excited to be speaking with you about some of the work I've been involved in over the past year today on the topic of intrathecal hydrocortisone for the treatment of children and young adults with CAR T-cell immune-effector cell-associated neurotoxicity syndrome. I am not cool enough to have any disclosures yet. So we've talked about chimeric antigen receptor, or CAR T-cell therapy, some already today, but light speed recap, it uses the immune cells from a patient and then genetically modifies them so that they can go in, detect, and destroy cancer cells when reinfused back into the patient. For example, in acute lymphoblastic leukemia, or ALL, the CAR T cells are made to target cell surface proteins that are specific to this type of cancer, such as CD19 and CD22. In this image here, we actually have a picture of CAR T cells found in the cerebrospinal fluid of a patient whose data we will be looking at later. And CAR T-cell therapy has demonstrated remarkable efficacy at inducing remission in children and young adults with B-cell malignancies, including those with relapse and refractory disease. And from what you guys have heard today, CAR T-cell therapy really is as cool as it sounds. However, it's not perfect, and treatment-limiting toxicities can result. The most common of these is cytokine release syndrome, or CRS. When this happens, the CAR T cells in the patient's existing immune system creates a hyperinflammatory response characterized by an overproduction of cytokines. This is very similar to how your immune system makes you sick when you have an infection, and this is also somewhat necessary to develop a response in patients. The second most common of these is immune-effector cell-associated neurotoxicity syndrome, easier said as ICANS, which we'll be talking about more today. ICANS is considered the black box warning label for CAR T-cell therapies, given the profound impact this neurotoxicity can have on patients. It is characterized by a pathologic process involving the central nervous system following any immune therapy, including CAR T-cells, and we see ICANS at rates of 28 to 50% of ALL patients after CAR, with 1 to 22% of patients developing severe ICANS. Clinical presentations of ICANS can vary a lot, including with mild patients developing difficulty writing, generating language, following commands, confusion, and delirium, to severe cases which may present with seizures, as seen in the CD19 CAR T-cell trials, encephalopathy, and cerebral edema, which can be fatal. Now, in the early CAR T-cell trials, there's no universal grading system, which led to the development of the 2018 ASTCT consensus guidelines depicted here. This grading scheme incorporates a composite score of encephalopathy, the ICE and CAP-D score for their respective ages, and symptoms in the 4 global domains of depressed consciousness, seizures, motor weakness, and raised intracranial pressure or cerebral edema, with the most severe symptom determining the patient's ultimate ICANS grade. Now, treatment for grade 1 ICANS consists of supportive care, including anti-epileptics and antipyretics, with patients with grade 2 or higher receiving systemic corticosteroids. However, second-line treatment is not standardized and generally at the discretion of the treating provider. This is a very critical area of research, as most patients who develop grade 3 ICANS will have persistent or progressive symptoms despite already being on systemic corticosteroids. Some therapies currently being tested in the field include siltuximab, an IL-6 binding monoclonal antibody that can enter the CSF, anakinra, an IL-1 receptor antagonist, and intrathecal hydrocortisone, which involves administering the steroid directly into the cerebrospinal fluid, most often through a lumbar puncture. At the NIH Pediatric Oncology branch, we have been incorporating intrathecal hydrocortisone into our treatment for grade 3 or higher ICANS patients, supported by the prevalence of intrathecal therapies in pediatric oncology practices in general, and the benefit of avoiding any additional corticosteroids in already vulnerable patients, or systemic corticosteroids in these vulnerable patients. There have been limited case reports of intrathecal hydrocortisone use with and without chemotherapy in adults with steroid refractory ICANS. However, there's even less, very limited data on its use in children and young adults. So, given the critical need to optimize therapy for ICANS, we completed a retrospective evaluation of patients treated on 2 phase 1 CAR T-cell trials held at the NIH. Data was restricted to those patients who went on to develop CRS, and these patients were separated further by the development of ICANS. ICANS patients were again stratified based on the severity, with mild defined as grade 1 or 2 ICANS and severe being grade 3 or higher. And also stratified by the receipt of intrathecal hydrocortisone. ASTCT ICANS grading was used, and any patient who predated this grading system was retroactively reconciled so we could look at all patients consistently. For this study, we also looked at patient cerebrospinal fluid studies, including white and red blood cell counts, protein, glucose, and CAR T-cell levels. These studies were collected from patients at baseline, after their CAR T-cell therapy at day 28 restaging, and also during the acute ICANS phase when available and possible. So we are going to digest this graph together a little bit. This figure shows here the breakdown of patients included in this review. Of the 79 patients enrolled on these trials, 53 of them developed CRS. 25 of these patients developed ICANS, and all 13 of these patients received supportive care as a treatment. 7 ICANS patients further developed severe ICANS, of which 6 received systemic corticosteroids as an additional treatment. 5 patients with severe ICANS received intrathecal hydrocortisone, with indications for use being grade 3 or higher ICANS, with the symptoms of progressive encephalopathy, gaze preference, focal monoparesis, and seizure-like activity. So, starting to look at our results a little bit, when we consider the cerebrospinal fluid studies, one concern when thinking of implementing intrathecal hydrocortisone is the risk of this steroid reducing the amount of CAR T cells in the cerebrospinal fluid and thus impairing the CAR T cells' ability to attack CNS disease. This work shows that when comparing our 5 patients who received intrathecal hydrocortisone with those ICANS patients who only received standard of care, the intrathecal hydrocortisone patients maintained higher level of CAR T cells at the CSF at day 28, indicating that at least in our limited number of patients, this treatment does not impact the prevalence of CAR T cell therapies, or of CAR T cells in the CSF. In line with prior research looking more closely at CSF studies in ICANS, we also saw that patients with severe ICANS had higher levels of protein in the CRS during the acute ICANS period as opposed to those who had mild ICANS. Other studies, including the white and red blood cell counts, glucose protein, and CAR T cell levels at their remaining time points did not differ. And this, really my favorite slide of this whole presentation, I think has the greatest takeaway here. Shows how really this is supposed to capture our experience so far that has led to our acquiring this therapy, showing that intrathecal hydrocortisone has led to a rapid and dramatic resolution of ICANS within 24 hours of receipt in all 5 of our patients. This figure depicts one patient's disease status and receipt of therapeutic attempts, with CRS grade shown in red and ICANS grade shown in black. And you can see how CRS responds to doses of tocilizumab and dexamethasone respectively, while ICANS continues to rise, only to fall rapidly after the administration of intrathecal hydrocortisone on day 5, even returning to a grade 0 after 2 days of receipt of this therapy. Also, all 5 of our patients did not require any additional escalation of care, any increased systemic steroids, or any additional doses of intrathecal hydrocortisone, and did not experience any reappearance of their symptoms. All patients were also able to be rapidly weaned from systemic corticosteroids, down to 1 milligram per kilogram per day of methylprednisone equivalent dosing within 1 to 7 days of receipt. In sum, our experience supports intrathecal hydrocortisone as a feasible and effective strategy for the treatment of persistent and progressive ICANS, with the benefit of rapid and dramatic resolution of symptoms, rapid wean from systemic corticosteroids without impacting the prevalence of CAR T cells at day 28 restaging. With a limited sample size of 5 patients, it is critical to pursue prospective studies to further elucidate the efficacy and feasibility of intrathecal hydrocortisone for the treatment of ICANS in children and young adults who receive CAR T cell therapy. And I'd like to close by briefly looking at how we use intrathecal hydrocortisone at the NCI Pediatric Oncology branch. And I would really like to emphasize how our experience to date has led to the incorporation of intrathecal hydrocortisone into our management of ICANS at the NIH POB. We implement single administration of intrathecal hydrocortisone using age-based children oncology group standard dosing at the development of grade 3 ICANS. We're using this as a preemptive strategy to prevent any further progression of their neurotoxicity, given the proclivity of these patients to progress while already being on high-dose systemic corticosteroids. And that concludes my presentation. Thanking everybody who I should thank would take 10 minutes on its own. So broad strokes, I'd like to thank everyone at the Pediatric Oncology branch, especially those in the hematological malignancy section. I would like to individually thank Dr. Haneen Chalabi for her mentorship, both in research and personally, and in advance for the support I know she will provide me in the future. Thank you to our NIH clinical care team who withstands all of the novel therapeutics and unique circumstances that our patients are in. Thank you to today's organizers and everyone at the Children's Cancer Foundation for this opportunity and a truly great experience for me as a up-and-coming researcher. And most of all, our pediatric oncology branch patients and families without which this research would not be possible. Thank you.
And Jim has a question.
Thank you. It was a great talk. I have a question about it. Seemed like, were you indicating that all the patients, I assume, were on systemic corticosteroids as well?
Who received intrathecal hydrocortisone? Yes, they were already on.
Do you know, were they on dexamethasone, hydrocortisone? Do you know what they were on?
I think they all received dexamethasone prior
Yeah, that
to
makes sense. Yeah, yes, I'm just curious if you have any thoughts on, because dexamethasone is highly brain penetrant, right, and it's more potent than hydrocortisone. So I'm curious, what do you think? Why do you think that intrathecal hydrocortisone is having such a significant impact when they're already on the systemic dexamethasone? Any ideas?
That is a great question. It's one that's been posed to us because, especially at high pulse doses of dexamethasone, this should penetrate the CSF at rates similar to what they would be getting with intrathecal hydrocortisone. In some, we don't really know why, and it's a really good question, and that's why I think prospective studies should also look at biological correlatives. This is kind of similar to how we adopted using tocilizumab in the early CAR T-cell trials, where it was what was available. This was something that was available and we thought could work, and we've just seen it to be very effective, even with patients already on high doses of dexamethasone. But that is a fantastic question.
Thanks.
Great presentation. I wanted to ask you if you, I'm assuming that dose of hydrocortisone is not that different than what we give with triples or something in leukemia, right? It's pretty low dose. So the question is, that's pretty benign. Nothing benign about doing an LP necessarily, but really we do them pretty routinely. Why not start earlier? Why not give it at grade 2? Why wait till they really start having more symptoms?
That is a great question, and I think that might be better addressed by some of our clinical practitioners who are here and may love to comment.
Great question. I think the neurotoxicity is probably, most patients don't progress to a grade 3, so doing LPs and potentially coagulopathic or thrombocytopenic patients are not as benign, especially in the acute toxicity period. But it is a great question. And it is standard triples dosing, so 10, 12, 15 milligrams. It's very low dose.
Yeah. And in addition, a lot of patients are experiencing ICANS during CRS, so they're not all necessarily stable. So to Dr. Chalabi's point, that lumbar puncture we want to avoid until we think it's absolutely necessary.
Trying to preemptively prevent toxicity, so not trying to predispose them to more procedures that they may not.
I was wondering, I don't know if you published this yet, but has the approach been, taken on by other academic institutions? I'm just curious because this is such a scary toxicity, and when you have a patient, then you probably would look into whatever can be done. Do you know if it has been, for example, DC Children's or Hopkins?
I don't know. Well, I know that we have published the limited case reports we have. Again, we have such a limited population of patients who actually go on to receive this. As far as I know, we are really the primary site that utilizes this therapy, and which is one of the reasons we're focusing on this work, and we want to look at prospective studies and biological studies to give support for other teams as well to begin utilizing this.
Thank you.
I just want to point out on the slide that the two physicians listed here, Dr. Shalabi and Shah, both were Children's Cancer Foundation Next Gen Award winners as well. So this is showing how they've kind of moved up and they have their trainees. So that's the goal. That's the goal of the program. And it's been so wonderful today we'll hear from you. I mean, this is great that you've got this experience. And I remember when I was finishing undergrad at Wisconsin, and I went to the International Society of Experimental Hematology, and I presented on the effects of gamma interferon on hematopoietic stem cells, and they introduced me to the platform presentation as Dr. Turetsky, and I was, 3 days into medical school at the time, so, a little preemptive, but a little premature, but it was, so congratulations, thank you. Okay. It's okay. I want to thank everybody for making it to the end of the day. And first of all, starting with Tasha, Lauren, and Roseanne,